Hello, I'm Brock Schumann, and I work at the Zebrafish Center for Advanced Drug Discovery in Toronto. Directed by Dr. Xiaoyan Wen, we use zebrafish as model organisms to study many human diseases with the end goal of trying to find new drugs to treat them. Why use zebrafish at all? Fish aren't even mammals, and people already do a lot of great research using rodents. With only a few arguable exceptions, fish are indeed less like people than mice are. However, they're still vertebrates, and as such, they have substantial genomic and physiological overlap with humans and are much less alien than invertebrate animal models such as C. elegans or Drosophila. They also have other properties, zebrafish, that make them highly amenable to truly high-throughput screens. A breeding pair of zebrafish lays a clutch of 200 eggs or more, which are easy to collect and a lot of material to work with. Because they're laid and develop externally, they're easily accessible for genetic manipulation, unlike mammals for which you have to extract fertilized eggs, manipulate them, and then reimplant them, each stage of which has some potential for failure. Zebrafish embryos develop very rapidly and will hatch into free-swimming individuals by the end of this 48-hour time-lapse video. Even by the 24-hour mark, the embryos have developed all their major physiological systems, and at only 2 millimeters in length, they fit quite comfortably into the wells of 96 well plates for assays or drug screens. For comparison's sake, a human or a rodent is only 8 cells by 72 hours, and mouse gestation takes 20 days. Humans is 9 months. Zebrafish respire aquatically, which is beneficial as drugs can be dosed directly through the aquaculture at concentrations in the same order of magnitude as are effective when injected. But probably the most remarkable feature of zebrafish embryos is their transparency. This allows for visualization of fluorescence and physiological processes in live animals that is just not possible in an opaque animal. These are some of the fish we work with in our lab to try and develop new drugs. But other groups have taken this fluorescence to even more extraordinary levels to visualize individual neurons or even individual action potentials using a calcium-sensitive fluorophore. One way to get meaningful fluorescence into a fish genome is using CRISPR technology, so we can design RNA that's complementary to a part of the fish genomic DNA and then translate the endonuclease Cas9, and it'll make a specific double-stranded cut just at that site. The embryo can then use endogenous repair machinery to insert foreign DNA. In this case, we'll insert the green fluorescent protein, and that green fluorescent protein will be translated under the control of the gene of interest endogenous promoter, and so we'll get tissue-specific expression of that GFP. This tissue specificity is useful in a drug screening context because if we're interested in increasing the fluorescence or getting drug activity in the brain, but not the heart, because, say, other drugs on the market for a condition have cardiovascular side effects, we can look for drugs that aren't active in the heart where those side effects are seen. The general protocol we use for our drug screens is to fill the wells of a 96 well plate with fluorescent reporter fish, expose each well to a different compound from a library of hundreds or thousands of chemicals, image and quantify the fish, and see any changes in the fluorescent that those compounds might have made. Our one-of-the-kind, high-throughput drug screening platform automates each of these steps, from arraying the eggs into the plates. Here, that's fish eggs on the right that are being dispensed into the 96 well plate on the left, to all liquid handling. And here is our robot. It's going to start with picking up 96 tips, and then it's going to pick up 96 drugs, and then dispense them to 96 fish. And it'll give them a little shake before putting it back into the incubator before this whole process culminates with confocal fluorescent microscopy of each well. So here we would be looking at each well, and while most wells would have the different drugs would make little change to the fluorescence of the fish, some of the compounds might have a different pattern of fluorescence in the fish, and so we might have found a new hit that we can investigate further. Because the images are confocal, they not only have higher resolution than traditional compound microscopes, they're three-dimensional images, allowing us to distinguish individual neurons or monitor specific blood vessels. So I hope you've enjoyed the video. Please feel free to either contact myself, Brock Schumann, or my supervisor, Dr. Xiaoyan Wen, if you are interested in collaboration, an interview, a tour of the facility, or if you have any questions